So, good morning, friends. Welcome to the last day of this colloquium. This is the 10th lecture in this colloquium, and we have 2 lectures in this session. So, today, Professor Chandrabas Narayana is here with us to give his lecture in this colloquium. First, let me thank Professor Chandrabas for accepting our invitation for delivering a lecture in the colloquium and addressing the students community to share his knowledge in spite of his busy schedule as the director of Rajiv Gandhi Center for Biotechnology, Trivandrum. And dear participants, RGCB is one of the outstanding biotechnology institutions in India. Now, let me introduce Professor Chandrabas Narayana. Professor Chandrabas obtained his PhD in physics from the Indian Institute of Science, Bangalore in 1995 and carried out postdoctoral research in material science in the Material Science and Engineering Department of Cornell University, New York from 1995 to 1998. He joined JNCSR as an assistant professor in 1998 and currently he is the director of Rajiv Gandhi Center for Biotechnology, Trivandrum, Kerala. Professor Narayana specializes in Raman spectroscopy. Though a trained condensed matter physicist, Professor Narayana developed a keen interest in biology within a few years at JNCSR. His group is pioneer in the use of Raman spectroscopy in drug-protein interaction for drug screening applications and diagnostic applications. Many of his work in this area are trendsetters in the interdisciplinary research. Among with his interest in biology, he also follows his interest in physics and chemistry. His group is one of the well-known groups in the country for use of Raman spectroscopy in the areas of physics, chemistry, and biology. His main research interests are magneto-Raman studies, temperature-dependent Raman studies to identify electronic, magnetic, and structural transitions, relevant spectroscopy for carbon nanotube and other novel systems and nanoparticles as biosensors. He is Fellow of Indian Academy of Sciences, Fellow of Royal Society of Chemistry, and Fellow of Indian National Academy of Sciences. So, dear participants, you are fortunate to have a professor like this because he has turned from physics to biologist, and he will show how physics and chemistry that has applications in biology. With this, now invite Professor Chandrabas for his lecture. Sir, please.
Yes. Thank you. First of all, let me thank Dr. Poti and Dr. Arunan for inviting me. And I am thankful for Dr. Raghu and Dr. Adish, both of them directors of CMET-Thrissur and CMET Hyderabad. They have been in touch with me and they are here, and I'm extremely happy to see them. And all the students, faculty, and our PhD students who are attending this. I'm very thankful for you to come on a Sunday and listen to this talk. I will actually make it very elementary or try to be very simple so that students of MSc also can understand what I'm talking about. So I would request the experts to excuse me for being too simplistic in my talk. So before I start, I would like to thank 2 of my students whose work I'm actually going to show here. That is Shantanu and Gayatri. Gayatri is currently in IIT Bombay, and Shantanu is now working in Renisha, India, which makes Raman spectrometers. So both of them have been the reason why I got quite a lot of work done in Raman and biology itself. So as topic is plasmonic enhancement of Raman scattering, which actually can be told as surface enhanced Raman scattering, and its applications. And mostly I will try to show the applications in biology because that is where the interesting things can be done with SIRS itself. Though there are many applications of SIRS which are there, it is not just biology, but I'm just trying to concentrate on that aspect. So before I start, I will actually start with the whole light-matter interaction, and all of us know that light actually does transmission, reflection, absorption, and then luminescence. These are the major activities which take place with light interactions. And many of the talks would have actually looked at many aspects of this itself. But what I wanted you to realize is that there is also other phenomena which are very, very low, less intense, and they are very interesting. One of them happens to be elastic scattering. And as the name suggests, it is elastic scattering. That means just like in which you have studied elastic collision, there is no change in energy. So that no change in energy means in terms of light, it means frequency doesn't change, and that means no change in color. So that's why elastic scattering happens in all directions compared to reflection or transmission, which happens in directionality is there. So this is one question I always ask students, but I cannot, because I'm not interacting with them. So I ask this question that how do we see each other? And most often students reply that we actually see each other because of the reflection. And I would like to point out that we do not see each other because of reflection. Of course, the colors, what we see is because of absorption of the white light, some part of the white light. But reflection is not the reason why we see each other. It is because of the elastic scattering. And I leave that as an exercise for students to find out why did I say this. In addition, there is something called inelastic scattering of light. That means inelastic collision means change in energy. The energy change in terms of light would mean color change. And I have only shown you a blue and red light, but it doesn't mean that the green light becomes blue or red. It is to signify that it gets blue shifted or red shifted. So that means lower energy or higher energy. So this is something which happens in inelastic scattering of light. And in fact, to show you that it does happen, I have taken an example of a liquid, which is here cyclohexane, which is a transparent liquid. And you can see when you put in a laser light, blue light, and you see that the blue light is scattered by the dust particle in the room. In a dark room, you can see that. And this is scattering because your camera is 90 degrees to the direction of the beam. You can see that even there is a strong scattering, elastic scattering happening from the liquid, much stronger than what is happening here. The reason why you see it as white here is because of the saturation of your detector. You know that whenever you take a photograph in a very bright sunlight, no colors are seen because the detector gets saturated. And that just because you know that there are 3 colored detectors, blue, green, and red. And if the colors which you see is basically the intensity of blue, green, and red varying. And that if it gets saturated, that means you have white light. That means all blue and green and red are equally intense, so it gives you white light. So this saturation is the reason why it happens. So that means you can see elastic scattering is pretty strong itself. And now when I put a filter which filters out a blue light, you can see that the elastic scattering of light from the dust particle has gone, and you can see a different colored light. And this is the inelastic scattered light. And this is the light which actually Raman saw, and he actually decided that we need to investigate. And he came out with the Raman scattering. This is actually a visible evidence of Raman scattering. So the reason why it is so intense is because it is organic liquids. And that's why Raman could discover this with a simple instrument. But today we use a very sophisticated instrument, and we need quite intense laser lights to actually do these experiments. So Raman spectroscopy is inelastic scattering of light, which actually looks at molecular vibrations. And in fact, these molecular vibrations are very unique, and they actually give you a lot of information about what is happening inside the system. And this is in inelastic scattering of light, as I said. So if you look at the energy diagram, you will realize that there are fluorescence and Raman actually use the same energy bands. So when you have ground state electronic states, they are coupled with the vibration, and this coupling with the vibration gives you these additional levels in the ground state of the electron. This is electron vibrational coupling. Now, when the light is incident on it, if it goes to an excited state, but it doesn't go into an excited state of the electron, but in intermediate state, then it comes down to the same state, then it is called elastic scattering. And this is called virtual states because the reason is these states are not real. These states happen when a molecule is excited. You have a linear combination of your eigenstates of electronic states. And that linear combination gives you, in an excited state, these states available for instantaneous time. So the light gets absorbed and instantaneously comes down, and that is elastic scattering. And that's the major thing which happens. But along with that, it may not come back to the ground state of the electron, but it may go to an excited state of electron with the vibration, and that can reduce the energy of the light coming out. That is the red shifted light. And along with that, you can also have blue shifted light, and that's what is called Stokes and anti-Stokes line. In general, Stokes is much stronger than the anti-Stokes because of the population. But you can also have excitation to an excited real state of the electron. And in that case, if the Raman happens, it is called resonant Raman. And at this, if this happens to be luminescent, then it also comes out with a fluorescence. So fluorescence requires luminescence associated with it, but Raman doesn't require a luminescence associated with it. If the excited state is there, it can actually access that if you have the right energy of light which you have incident. Raman is a very weak phenomena, and Raman actually can be seen as 1 in 10 to the power of 7 Rayleigh scattered photons. That means if you have a lot of elastic scattering, out of 10 to the power of 7 elastic scatterings, that means 10 million scatterings, which is elastic, only 1 Raman scattered happens. And now you will ask the question, how is that we are able to see Raman signal at all? The reason is Avogadro number. You all know that 1 mole of a material has 10 to the power of 23 molecules, and that 10 to the power of 23 molecules, each of these molecules scatter 1 Raman scattering, then it is 10 to the power of 23 scattering, right? So that's the reason why in a very large, in normal materials, you can see Raman scattering, even though it is very, very weak. But when it becomes very low concentration, then you start seeing that Raman is becoming difficult to see. And in the previous talk, yesterday, I think I think it is Sushil Majumdar or somebody had talked about surface plasmon resonance, and I am just going to recapitulate what he had said in a simpler way for me to continue with what I am going to do. So you all know that if I have a nanoparticle of a metal, then the metal nanoparticle can be defined as having a positive nucleus periodically arranged and a sea of electrons. It is a combined thing, and it is charged neutral. It doesn't have any charge, and it is actually a mixture of these 2 charges. But if I apply a perturbation, that means if I use a electric field, then I can actually displace the electrons because electrons are lighter, nucleus doesn't move. So basically, you will create a dipole. And that can be easily done by an electromagnetic radiation because it has an electric field which is oscillating. And hence, this charged dipole will oscillate. And all of us know an oscillating dipole produces electromagnetic radiation in turn. So that is why you can actually produce electromagnetic radiations from a light incident on a nanoparticle itself. And this is what is called surface plasmon. And this is surface plasmon because it is having more atoms on the surface and less atoms in the bulk in the case of a nanoparticle. That's why it's called surface plasmon. Otherwise, bulk also has a plasmon associated with it. Now you can see that if all lights are going to create a dipole, then why is there a color difference when the size of the nanoparticles change? You can see there is a wine red to violet color in the case of a gold nanoparticle itself, depending on the size. Now, this can be understood from the simple resonance experiments. You know that we have played this game of swing, where your intention is to pass your momentum completely to the swing so that the swing actually goes to large amplitude oscillations. So you would have realized that depending on the length of the swing, you would actually have to have a particular frequency of your motion of your hands to completely impart. If you are faster than that, you get hit, or if you are slower than that, you get hit. That is called resonance. And in exactly the same way, the oscillation of the electrons have a particular frequency, which is called the plasmon frequency. And if that frequency is matched with the frequency of the light, then you will have perfect change of momentum. That means you can exchange energy. You can exchange complete energy, and that means you will have a complete oscillation. If your frequency is too fast, then the electrons cannot follow it. It will go as if it is transmitted. And if the electrons, if the frequency is slower, then the electrons will actually reflect it. So there will be reflection. So if we now look at the photographs in transmission and reflection, you will realize the number of wavelengths which are reflected is different from the number of wavelengths which are transmitted in a white light. And that is the reason why a violet colored solution of large gold nanoparticle in reflection would look like brown, because of the difference in the reflection and transmission. So this tells you exactly which frequency of light is getting absorbed. So looking at that, we can know this is the resonance of it. Now, what people realized is that if I take a set of nanoparticles and put a molecule on top of it and incident a light, suddenly you see that very low concentration of sample also, the Raman signal is very, very intense. And this is what people discovered. And this is because of the surface plasmons which are present on the nanoparticles itself. And that is what we are going to understand. Why does the surge happen? So they actually came up with two mechanisms. One is called the electromagnetic enhancement. Another one is called the chemical enhancement. The electromagnetic enhancement is basically what I said. You create a dipole, and that dipole actually produces, oscillating dipole produces electromagnetic radiation. So actually the molecule sees not only the electromagnetic radiation of the light incident, but also an electromagnetic radiation of the dipole oscillating here. And again, there will be a cascading effect, because you would actually have Rayleigh scattering from the molecule, and that Rayleigh scattering again causes dipole movement, and then it is like a multiple cascading effect. And all of a sudden you see that the electric field which is incident on the molecule is going to be very, very large. And that's why you can get an enhancement of 10 to the power of 4 to 10 to the power of 8 in the case of electromagnetic enhancement. Now, what other thing happens is that if your molecule actually binds with a nanoparticle, then your Fermi level of the metal actually matches with the HOMO-LUMO mid gap. And that is normal. And hence you get additional channels for the electrons to go from HOMO to LUMO, and that increases the possibility of many electrons going into the HOMO-LUMO gap, and that means you will have more Raman signal coming from it. And this is called chemical enhancement, and that comes to about 10 to the power of 10 1 to 10 to the power of 2. That is basically. So you can actually have around 10 to the power of 6 to 10 to the power of 10 enhancement in general. And some of the cases, if you have resonance, then you can go up to 10 to the power of 14. And that is good enough to give single molecular detection also. So these are possible in search. Now, to show you that there is indeed chemical enhancement and also there is a enhancement due to electromagnetic radiation, the first discovery was done by a very well-known scientist in electrochemistry. His name is Fleischmann. He actually was looking at an electrochemical cell, and he was doing Raman on the electrode, silver electrode, with pyridine. And he realized that if you take a normal liquid pyridine, this is how the spectra looks. And if you take a very low concentration of pyridine, then about 0.05 molar, your signal reduces, but you can actually see the signal. But the moment you look at pyridine evolving around the electrode, he sees that the spectra is quite different from that. So selection rules are altered in the case of electrodes when pyridine is next to the sample. And now he did one more experiment, that he changed the electric field. That means he applied a voltage to the electrode, and all of a sudden he sees different spectra in the system depending on the voltage which he applies. And that was puzzling to him, and nobody could reproduce it. But later on, people realized that it was because of the nanostructuring of the silver electrode, which is not easy to produce in an electrochemical cell. But when you prepare a nanoparticle, you actually can reproduce this more often. And that's why now people don't use electrodes, but they use your nanoparticles for doing it. And that's why in 1980s, 84 or so, Van Dyne, who was the one who discovered the nano phenomena. So SIRS happens to be the first nano phenomena with which people have started looking at many applications in the current. Now, why does the surface plasmon resonance happen at a particular wavelength of a particular frequency of light? So you can see that when you have a nanoparticle and you have a wave pass through it, it produces an electric field around this system, and that is dependent on the dielectric of the medium around it and the dielectric of the metal itself. And this is because of the static polarizability of the electrons which are present in the system. And this is simple equations got from Jackson. You can actually see that, and you will realize that it has a factor in the denominator of the dielectric of the system, that is the nanoparticle, and the medium. Now, if I have air, then this is a dielectric of air is 1, and hence this is becoming epsilon plus 2 in the denominator. Now, interestingly, if you look at silver and gold, this is bulk material. You will see that at exactly around 3.1 in the case of 3.5 in the case of EV in the case of silver, and 2.7 in the case of gold, you have -2 as the epsilon 1, and that means this becomes 0, and that means this blows up. That is undefined. So that means you have a high polarizability. Now you realize the resonance. Why does it happen? Now, when you go to nanoparticles, this shifts towards lower energy, and that is the reason why we are able to do the search. Now, the enhancement factor is not very simple. It actually has enhancement factor because of the incident light, enhancement factor because of the scattered light, and all of these things is in a square. Along with that, there is also the distance between the molecule and the nanoparticle itself is a very important factor, and that's what happens. And you know that dipole mode is also there, and then is quadrupole mode. So both of these things actually act on the system. So that's why it's a very complicated way of electromagnetic enhancement which happens. So people have been interested in knowing how far can the molecule be. So one of the ways people actually did is by looking at DNA, which can be very exactly measured. How much is the distance between the molecule and the gold nanoparticle? So they realized, and they did this experiment, and they found that it can actually go up to 15 nanometers or 10 nanometers you can actually detect, which was unusual. People couldn't realize. So people then went, instead of DNA, they actually went to methylene spacers, that means CH2, CH2, CH2. And again, they found that this was giving you around 10 to 12 nanometers as the length to which the molecule can be kept. But the problem with these two spacers is that they are not rigid. They cannot be stiffly standing. They can actually buckle. And because of as the length increases, the buckling will be more. And hence, you may not be actually measuring the right distances. So people did another experiment where they put a polymer coating on top of this and tried to measure the Raman of the polymer. But as you know, polymer also doesn't give you a clear idea of how much is your extent of this thing. And in here in ISA Trivandrum, George Thomas group actually tried to find out how this distance phenomena can be actually done. So what they did is they coated the nanoparticle with silica of different thickness, added a dye molecule on top of it, and then tried to do a functionalization so that the dye molecule actually forms a linkage. So these two dye molecules will come together to produce a dye in between the two systems. So they changed this, and they found that it was very, very close to around 2 to 3 nanometers is the maximum detection possible. Now, again, here there is a problem because this dye is also a issue when it comes to doing these things. So what we did is, we said we will not use any functionalization. What we will do is we will vary the thickness of the system. And when we change the thickness of the system by silica coating, very special coating, so we can control about 1, 2, 3, 4, 5 nanometers. You can see the thickness increases. Now, if I use any dye on top of it, I will get the source of that. So we also did one more thing. We also changed the size of the nanoparticle. We went from 30 nanometers to 90 nanometers and to study what happens in the case of the system. Now you can see 30 nanometers, 45 nanometers, 75 nanometers, and 90 nanometers. We use the same concentration of R6G, and you can see to what thickness you are able to detect this Raman signal. As you can see, as you increase the size of the nanoparticle, you are able to go to thicker and thicker SiO2. And if you plot it in a graph, you can see that if you have a smaller nanoparticle, the detection level is about 2.5. But as you increase the nanoparticle, your detection level keeps on increasing. And for 90 nanometer silver nanoparticles, you can even detect up to 5 nanometers. So that is what is more realistic, and people have actually now understood that it is indeed very, very important that what is the size of the nanoparticle also matters. And if you do a calculation by FTTD, you can see that why it happens. When you have a smaller nanoparticle, the effect, plasmonic effect, is electromagnetic distances is very small. As you increase the nanoparticle, the distance actually increases. And you can see that matches exactly what we had actually measured in our system. So when we do this with biological system, we can do a very interesting set of problems. And now next few examples will be based on how Raman can actually help you in doing various experiments. But before we go to the examples of application, we would also like to show you how we make new nanoparticle structures for doing these experiments. So whenever we are doing biomolecular system, what we do is we incubate the biomolecule with the nanoparticles and keep it in the microscope in the liquid itself. Otherwise, the protein will denature. So this is the kind of concentrations which we use. And typically, we use about 30 to 3 minutes as the accumulation time. And our laser powers, we try to keep it as low as possible, below 5 milliwatts. We can use a commercial Raman setup, and you can use multiple wavelengths of light, or you can build your own setup. And that's what we have also done, because if you want to take it to application, you have to build your own system. So here is a built system where you can actually have a 96 well plate of the biological systems, and you can do a scanning on top of it, and it is kind of handheld system where which has been immobilized here, and we can actually do a very beautiful analysis of diagnostic applications for that. This is the architecture of it. And you can here see that most of our experiments with protein, we use nanoparticles of silver, which have a size varying around 50 to 60 nanometers. And that's what most of the times we use. And this is citrate reduced, because citrate is easy to dislodge by the protein. So that is why it has a charge dependence. So this is negatively charged. So most often the protein actually comes close to the nanoparticle with the positive charge. So the positive charge is very important for it's to absorb onto the silver nanoparticle. Now I will show you that we can actually, one of the major problem with the nanoparticles is that they have a charge dependence. Now you want to make it charge neutral so that any kind of molecule can be tested, and that's where we were actually investigating, and we found that there are very interesting effect of dielectrics on the nanoparticles. And that dielectric, we had discovered that while we were working with gallium nitride and other systems. So suddenly, I got a collaborator in National Aeronautical Laboratory, Harish Barsilia, who actually works on trying to make a good solar photovoltaic cells. And he's been working with silicon to make the reflectivity of the silicon very low. And so he started nanostructuring the silicon so that he can have very low reflections in the visible range. And that is what he was working. And then he realized that there is a very interesting combination we can do with me to do a surface enhanced Raman spectroscopy. So what we realized is that we can, colloidal solutions have limitations, but solid supports have a much better application. So we came out with two different types of nanostructuring of silicon. And one is called nanocement. I will actually show you that. Another one is nanosilicon pillars. Now what we did is, he used to make these nice pyramids of silicon by etching, which is very scalable process. And on top of it, we deposited a silicon nitride on it. And then we did silver sputtering on top of it to make nanoparticles of silver. And this gets immobilized. And you can actually control the size of the nanoparticles. And this size of the nanoparticles will actually give you what kind of effect we see. So you can see this is how the nano pyramids are created. And these pyramids have been deposited with silicon nitride as well as silver. And you can see very nicely, you can control it. So you can actually control the size of the nanoparticles by sputtering, film thickness, and roughness. All of these things can be controlled. But interestingly, what we see is that we actually get a very strong enhancement from these systems where the enhancement factor is about 10 to the power 6 to 10 to the power 7 easily. So you can control that. And also we find that higher sputtering power and film thickness gives you very good enhancement properties. The interesting effect is that it is independent of the wavelength of light. So we could actually do it with 532 and 633 with equally giving you very good enhancement for most of our sample. But we were not satisfied because it was giving only in the visible range. What we wanted is Raman should be in all ranges. So we went ahead and did another experiment where he had got a nice nano pillars of silicon by he was doing etching processes, and it is metal assisted system. So you can control the size of the nanoparticles and nanopillars of the silicon, and on which we spetter coated the silver particles on top of it. And we can control the size of the silver nanoparticles. But the interesting thing is that the plasmon actually became not localized. It has actually surface plasmon was active in all ranges of the wavelength of light. And this was very interesting for us, because that means we do not have any dependence on a particular wavelength of light we need to work with. Now you can see we use UV light, we use red light, and we use infrared light. So the enhancement is more or less consistent with the system, and it is roughly around 10 to the power of 8, which is good enough for most of the biological systems. And the advantage we have is it is reusable. So if you put a molecule on top of it, any organic molecule, if you heat it up to 400 degrees, it actually disappears. So you can actually reuse it multiple times. Or it is also not having any effect of corrosion. You can also use HCl to clean up the surface. And you can see that it repeatedly gives you much better enhancement in the system. Now, to show that it is a multi-analyte system, both of these systems, whichever we have shown, is a multi-analyte system, because there is no capping agent in this system. And that is why it can detect dyes, it can detect proteins, lysosomes, BSA, it can detect ATP, which is a energy packet, also DNA. All of these things can be detected, and you can see it can detect at a very low concentrations, about 10 to the power of minus 12 to 10 to the power of minus 13 molar. We can actually detect this dye or a thiophenol in the system. And interestingly, you can even put a cell on top of it. And here is a RBC, red blood corpuscle. And you know that if you want to find out you have malaria, that's the RBC will be infected with the malarial parasite, and hence its Raman spectra is different. So you can see the difference between a normal spectra of a RBC and an infected RBC. You can easily find the distinguishing factors, and you can distinguish which cell is, whether the malaria is there in the system. Even you can detect bacteria on top of it. It is an E. coli which has been put on top of it, and you can see the presence of E. coli. And in fact, any dye which is used for detecting various fluorescent molecules which are used in biology, we can actually detect using this. So this basically becomes a very good tool for measuring any type of systems. So you can see why the enhancement factor is there. FDD calculations clearly show why we have a very strong enhancement in the system. I will go to the next example, where I'll show you two examples of use of SIRS in biology, where we address very important questions which are very difficult to do in normal sense. And there are very tough experiments to be done in most of the cases, but SIRS becomes an easy way to look at it. And any proteins are actually made of amino acids, but these amino acids actually combine together by peptide bonds, and these peptide bonds are nothing but CH2NH. This is what is a C double bond O NH is the peptide bond, and it has a possibility to do hydrogen bonding. And there are many different types of hydrogen bonding it does protein, and that is what is called the secondary structure. And the secondary structure gives them alpha helix, beta sheets, or random coil. That means it is open structure. So any of these things can be present in this, but you have a very interesting vibrations associated with the C double bond O and NH, because C double bond O stretching frequency and NH bending frequency in plane is exactly matching. So it is like two harmonic oscillators which are having equal vibrational frequency, and whenever they are coupled together, they would actually cause multiple vibrations, and that's called forced oscillations. And this is what happens. You have amide 1, amide 2, and amide 3 because of the different types of vibrations possible in the system. And this is very important because it is dependent on the hydrogen bonding which is present, and that is going to be easy to detect. So if you take any protein, the protein's amide 1 region is easily distinguishable into beta sheets, alpha helix, and random coils. And you can actually see from statistics, you know exactly where they collapse onto. And so you can actually detect these. So with this tool in hand, what we did recently, and we have been working on this for a long time, but this is one recent example where we were actually trying to understand how does a divalent metal ion change the structure of the protein to do structure, because protein's function depends on the structure. So structure function relationship can be understood. So how does it modulate it? This is the thing which we wanted to understand. So we were working with a restriction enzyme. Restriction enzyme is an enzyme which cuts DNA at specific sites. And this is present in most of our systems like bacteria. So generally the restriction enzymes are named by the bacteria in which it is there. This is the Calabacillia pneumoniae. So that is where the Kp comes from. And N comes from the pneumonia, Pn, next number. And it gives, the Roman numbers are given as 1, 2, 3. So it's called Kpn1, is one of the restriction enzymes. Why is it very interesting? Because this bacteria protects itself it's a defense mechanism to cut the virus which attacks this bacteria. So it actually slices the DNA of the bacteria by recognizing the specific DNA sequence. And this DNA sequence, whenever it finds, it will actually cut it. And once the DNA is cut, this particular virus becomes inactive. And so you know now that how we can use this mechanism to control our viruses which attack us. So this restriction enzyme study is a very important thing. Now, what they also found out is that if you change the metal ion in that, because these restriction enzymes are having metal ions to get the tertiary structure, to put them together in a folded state. And that is very much dependent on what kind of metal ions are present. Now, what they found out is that if you put calcium and if you put magnesium, the structure is quite different. And if the structure is different, its property is different. So you can see that if you have calcium, this actually recognizes this sequence very, very precisely. And if the magnesium is put, if we put magnesium in smaller quantity, it recognizes this series. But the moment you put higher concentration of magnesium, it becomes promiscuous. That means it doesn't look at only this sequence, it cuts anywhere and everywhere. So promiscuity increases when you have a magnesium. So just the difference of a cation, it changes. And it was known that this cation, not that the structure of this particular protein is known, but at least we know where the metal ion may bind. And based on that, they have found that it is actually binding in two regions, HNN and EDT region. And that is the expansion for all the amino acids which is there. So these two regions are the regions very important for binding of this metal ion. And we wanted to understand how does the calcium and magnesium bind differently to create different binding. So crystal structure not being known is a very difficult thing to work with. And we wanted to know if we could exploit this defense mechanism to gain fitness advantage over a host. So this is something which was the question we were asking. Now, whenever we don't know the crystal structure, we look at the amino acid sequence, and then you can actually model it by homology model. And using the homology model, we have modeled the crystal structure of the wild type without the nanoparticle. Without the cation. And when you don't have the cation, you can see there is always a zinc, which is very, very important in our most of the proteins. This is called the zinc folding. It actually gives you a structure like this. And this is the structure of the EDD, which is predominantly a beta sheet. And the HNH region is mostly predominantly in the region of beta sheets and alpha helix. So this is what is the situation. And we could actually see that where it binds in the system. And with this in hand, we could actually look at the charge, because we wanted to understand how does it go and sit on top of the nanoparticle. So if you look at the charge, we see that around this region, there are two reasons for it to attach. And by the way, this protein exists in a dimeric form. That means it is not one protein. It is two of the proteins coming together and actually making into a protein which is viable. And this is the dimerization of that. And you can see there is a lysine group sitting here. That is the anchoring point. And this is the region where the most positive charges are there. So this is how it would be sitting on the nanoparticles. Once we know that, when we do the Raman spectra, and we can see that most of the low frequency modes are associated with aromatic amino acids, but the high frequency region around 1600 is because of the amide region. And I have color coded the beta sheets and alpha helix. And this is the dimer of that. And you can see that this is the beta sheet, which is coming closer because this is the EDTD region. And this is the alpha helix region, and that's what is seen. Now, when we put the magnesium into it and compare the spectra, you see that the amide region doesn't change much. So there is no change in the amide region. So wild type is this. When you put the magnesium, we did the calculation, and we found that there is no change in the EDD region. So the magnesium binding into the EDD doesn't change the system. And this is the reason why these two behave promiscuously, because the structure function is similar to the wild type itself. So we could actually understand that. Now, when we put the calcium, we immediately saw a new peak coming around 1680 and above, 1670 and above, which is due to random coil. So that means some of the beta sheets are getting converted into random coil because the intensity of the beta sheet goes down and the intensity increases, which shows that some beta sheets are getting converted. Now, when we did the modeling, we see that in the EDD region, which is a beta sheet rich region, that actually becomes, when calcium is bound, becomes a random coil. And that is the reason why we have a very specific folding. And that specific folding is actually the reason why calcium is doing a specific recognition. And in fact, this is the homology model of that MD simulation. And you can superimpose the whole structure, and you can see the wild type, which is given by the color cyan color. And green is the calcium bound, and magnesium is purple. And when we superimpose it, you can see the wild type and the magnesium do not change this beta sheets, but the calcium actually changes, and because of which there is a subtle variation in the tertiary structure of the protein. And that is the reason why this is able to do a non-promiscuous binding. And so that is basically what so we could actually do this. Another one very important thing which we can do with the SIRS is for drug screening. Drug screening is a very important thing in biology, which we started working with Guru Rao in IISc. And Guru Rao was working with medical companies which were interested in finding out drugs for potential applications. We were working with kinases. And what is a protein kinase? Protein kinase is an enzyme which actually works on any protein, and it actually phosphorylates it. That means it takes a ATP and converts it into ADP, and that one phosphate group is attached to it. And this phosphorylation is a very important process in cell control of various activities. Now, what kinase we were working is C-Jun N-terminal kinase, that is called JNK, because it phosphorylates C-Jun protein at the N-terminal. And that's how the name comes, C-Jun N-terminal kinase. And this kinase exists in three structures, JNK 1, 2, 3. And all of these three kinases are very important. And they are very much dependent on the stresses applied on the cell. And based on the stresses, they can have abnormality in the phosphorylation of C-Jun. And once it is abnormal behavior is started, it can cause many diseases like neurodegenerative diseases, chronic inflammation, birth defects, cancer, ischemia, that is heart diseases, obesity, insulin-resistant diabetes, and many others. So it is very important to inhibit this phosphorylation of C-Jun in those patients where the JNK is phosphorylating abnormally. So inhibition, when we talk about, this is a simple lock and key mechanism. So this is the substrate and this is your enzyme, and enzyme actually binds it. So you can actually inhibit it in two different places. One is competitive inhibition. That means it competes with the substrate and binds to the active site, or it binds in some other place and changes the size of the active site. And that can make it difficult for the substrate to bind. So this is called competitive, and this is called a non-competitive inhibitor. So it is found that this particular molecule, 1,9-pyrazolanthrone, is a inhibitor for the C-Jun kinase, JNK molecule. So this was actually a good inhibitor, but not very good, because if you are using it in large quantities, it can have side effects. So people are interested in making this molecule better, and so people started looking at many derivatives of it, and that's what was the reason why our Guru Rao was interested. He is an X-ray crystallographer, so he was interested in finding out how this particular molecule is binding to the system. So the kind of changes what was done is that we increase the hydrophilicity in this region by alkyl chain plus NH2 and adding a chlorine region, which is a hydrophobic region. So you change this and you make many new molecules, and you can see different types of molecules were created. So they were interested in knowing which of these things can be quickly understood as a possible candidate for enhancement, inhibitory enhancement. So that is what they were interested in. So they came to us. So usual method is you synthesize new molecules, do the biochemistry, then do the mouse model, and then the X-ray crystal structure. But that is a very exhaustive way, because if you want to do for all the molecules, what we would like to do is first you synthesize the new molecules and see if it is really working. So you can use SIRS to eliminate the unwanted molecules. And once that is done, those molecules only you need to study. So that is the reason why SIRS becomes a very important tool. So what we were interested in is, can we do this? So if you look at the structural thing of JNK1 and JNK3, if you superimpose the two structures, you can see very little difference is there between the two. 99% they are 92% they are similar. You can see that the electrostatic map is also looking very, very similar, but there are differences in the electromagnetic. So here is the sequence alignment. You can see 92% sequence identity is there, but you can see the differences are in the alpha helix, beta sheets, and you are having single nucleotide changes happening in the system. So those are the things which makes them different. Otherwise, they both are identical. So what we wanted to know is, which can we quickly look at the screening of this molecule. So we understood that the molecule is binding to this region, and this region happens to be within 1.7 nanometers. So naturally, this is an organic molecule and this is a protein. And if the organic molecule, though it is an insoluble in water, but this silver is in aqueous solution, but since it gets localized here, we know that the organic molecular signature will be much higher. So if the organic molecule is higher, if this is bound well, the signal of the system will be much more. So we did the JNK protein and JNK 1 and 3 protein spectra. So we know the spectra of the JNK. What we did is we put the inhibitors at different points, and what we looked at is the intensity of these. These are all normalized Raman spectra. So you can see RAN2 has the highest intensity, which means RAN2 binds much more strongly to the system compared to even the SP, which is the original molecule. You can see. So you have possibility of, based on this, I can actually tell which binds better. So RAN2 is this molecule, which binds best for the JNK. It is much better than the even the initial molecule which started. And this is the ranking of the different molecules. So you know which is the best molecule. So you can rank. In fact, we checked this with the biochemical assays, and we found that 1-to-1 correspondence in there. Now, when we put it in JNK3, even here, the RAN2 binds much better compared to the SP, but it is not very different. You can see RAN2 is 182 here, and SP is 160. So it is more or less similar. So you can see that you can rank it, and this ranking is exactly matching with the biochemistry of the system. So what we are showing is that by just looking at this Raman, you can immediately know which molecules you need to work with, because all the molecules which work better than the original molecule is the molecules you need to work, and hence we have localized that RAN2 is the better molecule to work with. Now, to show you that it binds differently in JNK1 and JNK2, RAN2 molecule is this. You can see the Raman spectra is more or less similar, but there is a very subtle variation in the intensity of these two peaks. And what does it mean? Is because this particular molecule is flipped. It is actually getting flipped. And that is the flipping of this molecule gives you this change in the intensity of this. And now if you look at the molecular modeling, you also see that your NH2 is facing towards you in this particular JNK1, while NH2 is facing inwards in the case of JNK3. So this pocket, though happens to be very, very similar, but the interactions with the protein is quite different, making this molecule to sit differently. So you can even not only look at the screening, but also tell the homology model is working very well. So it can actually be a very good tool to verify binding of the molecules also. With that, I come to the end of the talk. I actually have shown two applications of it. One is to understand the structure function. Another is for screening tool in proteins. Many of these things are very, very important, and we can do it with this. Thank you for your attention. I hope I have completed in right time for this talk. Thank you. Thank you.